Viruses like coronaviruses are what we call envelope viruses, because when they emerge from the cells that they are infecting, they steal a piece of the surface of that cell. And so if you think about it, the parts of the virus that tell its host cell how to make more viruses is trapped behind that envelope. And so once this virus needs to go into a new cell, it needs to somehow get rid of that envelope. And it does that by fusing with either the surface or one of the internal envelopes inside the cell. And then the genome can escape, the information can escape into the cell, and the cell starts making new viruses. So what we are trying to do is tackle essential steps in the life cycle of a virus and block the ability of the virus to infect. We actually have expertise in all of these steps, how a virus might bind to a cell, how the cells might take it in, how then the envelope of the virus and of the cell fuse to each other. The tools existed in the lab, so it was not a difficult transition. The reason coronavirus is called coronavirus is because it has this crown. This crown is made out of a protein called the spike. The spike protein is the protein that mediates all of these steps. So that's the protein that we are putting on our pseudovirus. You can imagine that working with a real life virus can be quite challenging. So what we are generating is a pseudovirus. It's a virus that will carry on its envelope the key protein, the key component that mediates these steps. However, this virus is not able to replicate. And instead of making new viruses, the cells start emitting light. And so by screening for either cells that emit light, which are infected, and cells that don't emit light, which haven't become infected, we can distinguish, for example, compounds that inhibit, that block the ability of the virus to infect. Because if we see cells that don't light up after we add our virus, it means we've blocked one of the essential steps of infection. The reason we chose this strategy is that because this step of the infection cycle is the first right at the beginning of infection, which means that if we are able to block that step, no infection will happen. This is a step before immune responses, before where a vaccine would be relevant. So we are currently at the stage where we should, next week should have this attenuated, this lab adapted virus. And then what we can already do is we can grow cells in the lab. These are cells that are derived from lungs. So they have all the receptors, everything that, our lung cells would have. And they're the perfect sort of medium to test whether our virus can mimic the infection of corona. We are in touch with a few both institutes and other researchers on campus that can provide us with different compounds from plants, from FDA-approved libraries that we can screen whether those can inhibit viral infection.